So, transcription, the first stage in protein synthesis. If you ever need to remember which one comes first, transcription comes before translation, because alphabetically that would happen as well. So trans C, trans L. So transcription is just a fancy word for RNA synthesis. If you think about it, it's RNA synthesis from a DNA template. So all we are doing when doing transcription is RNA synthesis. So we're going to use our RNA polymerase because RNA is a polymer of RNA nucleotides, which we've learned about earlier on. So how does it work? Well, the DNA gets unzipped, which we'll talk about later, and once it's unzipped you get exposed bases. You get these exposed bases. And what ends up happening is, just through sheer diffusion and things bouncing around, eventually an RNA U nucleotide, a uracil nucleotide, will form two hydrogen bonds with this A, and it will then sort of slot in next to it. And then, just like the DNA polymerase, RNA polymerase can only grow at the 3' end. It will then form a phosphate sugar backbone from the 3' carbon to a phosphate. So you reform the phosphate sugar backbone. This is kind of important. So the nucleotides come in as triphosphatidic nucleotides. So ATP, GTP, CTP, whatever you want. The energy for transcription comes from breaking off these phosphate groups. You know that the breakdown of ATP releases energy. It's the same with GTP and CTP. This is an energy requiring process. So you use the energy from breaking off the P's, the phosphates from these nucleotides to give you energy. And that just carries on. It just goes bobs along. And then, I don't know, an A will come in here. So literally ATP will come in. And then it will form a hydrogen bond, form a covalent bond, a phosphodiester bond to this ribose, and you get a phosphate up here, and then it will continue and continue and continue. You've got RNA synthesis, haven't you? However, slightly more complicated. So I said that you need to unzip the DNA to expose those bases. It's actually far easier than DNA synthesis because RNA polymerase has a helicase function. So if it needs to transcribe this strand, what it does is it just unzips it itself and then grows in the 5' to the 3' end. And that's it. If it needs to go the other way, all it does is the RNA polymerase flips itself over. So it actually unzips it, and then you can form a 5' end here. So it just flips itself over, and then that's now the 5' end. This is the 3' end, and because it's got its own helicase, it just goes whoop the other way. So actually, RNA polymerase and synthesis is much easier than DNA synthesis. The one thing you do need to be aware of is this idea of which one's the coding strand in DNA and which one's the template strand. So we have unzipped this area, so we've broken the hydrogen bonds between these two things. There should be two hydrogen bonds here, 200 bonds here. And what we've done is we've started making this RNA strand. The strand that is acting for the template for the RNA nucleotides to come in is the template strand, as suggested. And the coding strand of DNA, so that's the template strand of DNA, the coding strand of DNA is the one which has a very similar code to the one that is on the RNA. So they code for the same things. So apart from the U's, you've still got A, T, G, A, T, G, A, T, C, and then that would be a T, that would be a C, that would be that would be a U, sorry. That would be a C, that would be a G, that would be a U, that would be an A, and that would be an A. So the coding strand is actually the one which has the very similar code to the one on the piece of mRNA.